Hello everyone, and thank you for attending today's webinar, How to Apply Microanalysis to the Life Sciences. Before we begin, I would like to go over a few housekeeping items. At the bottom of your console, there are several application widgets you can use. If you have any questions during the webcast, you can click on the Q&A widget and submit your question. We will try to answer any questions during the webcast, but if a fuller answer is needed or if we run out of time, you will receive an answer later via email. A copy of today's slide deck and some links to additional materials are available in the resource list widget, which looks like a green folder. You can expand your slide area by clicking on the maximize icon on the top right or by dragging the bottom right corner of the slide area. Finally, an on-demand version of the webcast will be available shortly after the live session, and it can be accessed during the same audience link that was sent out to you earlier. Hello, I'm Microanalysis. I'm the Global Applications Manager, and I'm really excited to talk about life sciences and microanalysis today. So microanalysis and life sciences don't often go hand in hand. In fact, there are much fewer people in the biological and life sciences who actually know the benefits and use microanalysis to help solve their problem. And what we're here to offer today is a chance to understand at a deeper level how you can apply microanalysis to solve some of the life science challenges. To get started, I'd like to start with a little quiz or a survey. This is just a little bit of fun here. Recently, over the summer, we had a conference, the Microscopy and Microanalysis Conference, and I had done some work and came across this very interesting sample. So I took a very low mag image of it, and it is rather large in the world of microscopy. It's a 2-millimeter feature. And I thought it was very interesting to look at. So I'd like to ask everybody to take a moment to take the poll following this slide to see what you think it is. So I'll give you a couple of hints. It is something that reminds us of this time of year, the autumn, at least here in the U.S. It's celebrated with some fall foods and, for example, pumpkin pie. So it's something that you would encounter during the course of your preparations for the upcoming season. And a few of the options are here. Just simply click on any one of the selections that you believe that image might be. So what is this autumn ingredient? And just take a moment to review the options: pomegranate, spice clove, acorn squash, or an alien life form, which it does kind of look like. So we'll give another couple of seconds for you to make a final choice, and then we'll move on to see the poll results. Okay, we've got a lot of great guesses, and over half of you have got it correctly. It is, in fact, a spice clove. It is a full spice clove that was one of the outer shell casings was peeled off, so we looked inside with the electron microscope and were able to see the components of the clove. And so over half of you got that right, which is great, and I was super happy to see that there was at least one person who liked the gag or joke question, so great to see the alien life form. People are having some fun with it. So today's webinar is going to be a little bit different than our webinars. Usually we talk about some really deep topics of detector electronics and speed and how we convert X-rays into energies that we interpret in a spectrum. Today we're really just going to take some time to explore some of the fascinating applications of microanalysis in the life sciences. So as somebody who has been studying biology from early on and fell in love with microscopy from even before going off to undergraduate college, I really enjoy finding different ways of looking at biological materials in the microscope and taking it even further with some of the microanalysis tools available to us today. So why are we at such a good place in the point in the technology time? There have been some revolutionary changes in the last 3 years that have improved EDS detector performance that really opens up the world of biological and life science applications wide open. And the most important change that we've had recently is the introduction and common use of the silicon nitride window in our silicon drift detectors. This was introduced about 3 years ago, and the benefit is that it provides 2 to 3 times more sensitivity for nitrogen and some of the low energy X-rays compared to the previous generation of polymer windows, which were around for decades. The silicon nitride window is much thinner, and so it allows better transmission for those low energy X-rays to get through the window, and so it improves our detectability and also increases our ability to solve challenges in the life sciences. What we also pair it with is high efficiency electronics. And those high-speed electronics maximize the signal processing, even at low kV or low beam currents, low count rates, which are typical of life sciences application. So even if you don't have high count rates, you could still reap the benefit of high-speed electronics by keeping your dead time low, by being able to make the most of the few X-ray counts that you have. And finally, we pair with some fast mapping processing enhancements, and that even if you have low signal, we're able to amplify and increase the signal that you see in your X-ray maps so that you're able to get nice high-resolution images or small feature sizes, even of those challenging low-energy X-rays. All at the same time, we're able to do this quickly. So there's no longer the need to dwell on your sample for many minutes or hours, which, as we know, can damage a sensitive tissue sample or seed sample. So nowadays, we can still preserve the integrity of our biological or soft tissue materials. To get started, I will show a spectrum that emphasizes 2 of the main characteristics that are useful in the life sciences. Those are sensitivity and resolution. And for those 2 terms, we're referring to the spectral sensitivity and peak resolution. Sensitivity, it allows the peak to stand above the background. So even in a low signal environment, low count rate, low kV, low X-rays, low energy, you still have the ability to pull those X-rays, those peaks, out from the noise, or the signal above the noise, and the result is your spectra that has a peak above the background. And you can see in the image that we have here of the spectral overlay, we have some trace amounts that are just slightly above the background, but with the improved sensitivity of the silicon nitride window, we're better able to see the peak above background. Now this is a human hair sample, which makes a readily available and easy to use sample to put into the microscope. It's a great way of looking at detector performance, and everybody has, or I think most everybody has, availability to such a type of sample that you can easily put into the microscope. And this spectra shows your carbon, nitrogen, and oxygen peaks. The important things are that we're pulling the peaks out of the background. Most importantly is that we're getting that better sensitivity in nitrogen area with the silicon nitride detector. And that's that peak in the middle of those 3 peaks there. With the silicon nitride window in outline, you could see the great improvement of nitrogen over the polymer window nitrogen peak. The reason for this is because the polymer window has carbon in it, and carbon has an X-ray absorption edge just higher than the carbon peak energy, which falls exactly on nitrogen. And so it means that the polymer or super ultra thin windows are highly absorptive of the nitrogen signal. And so in times past, it would always be a challenge to be able to determine low nitrogen levels. Nowadays, with the silicon nitride window, we're able to pull that peak out of the background. We're able to eliminate the absorption edge because there's no carbon in the window, and then we're able to get the high nitrogen sensitivity. One of the comments is usually, well, you have a silicon nitride window, does that absorb anything? And the answer is not much at all. And the reason for that is because the silicon nitride window is about 7 times thinner than the polymer window, and so there is very little interaction between the X-ray and the silicon nitride window, so we don't get some of those artifacts that we used to get as well. So sensitivity is important because it allows us to detect low concentrations of material and also trace detections, and we could do it more quickly as well. In biological materials, we'll often find discrete particulates or areas that are different than other areas. And when you're doing a scan of a large area, but you only have a small contribution from one feature of interest, that feature signal is going to be very small. So the better that we can get a high sensitivity detector, the more likely that we're able to detect and find that feature in our spectrum and our maps. Now, the next term that we'll talk about is resolution, and this is one that we commonly hear in the world of EDS detector performance. In fact, we hear resolution a lot in microscopy, and they do mean slightly different things depending upon how we use them. So in the EDS world, resolution means the ability to distinguish two peaks from each other, 2 or more peaks from each other. And these are usually neighboring peaks. So for example, the carbon, nitrogen, and oxygen are in sequence. And so the ability to tell the difference between the carbon, nitrogen, and oxygen peaks is very important because then we could determine what elements are present in our sample. And also equally important in spectral analysis is we like to see the peaks go back down to the baseline or as far down to the baseline as we can. And that gives us our nice ability to separate those peaks. So there is a tie-in between sensitivity and resolution because the better that we're able to pull the peak above the background with better sensitivity, the better we're able to separate it from the neighboring elements. Likewise, on the flip side, the better resolution that we have, the fewer channels it bins the signal into, and that gives us the ability to get better sensitivity just simply by nature of binning the X-ray into the proper channels where it belongs. So now the second of 3 surveys. So we'll go through just a couple surveys pretty quickly, but we'd like to increase interaction with the audience in our webinars. So I'd like to ask all of you, what functions of your detector technology are most important to you? So do you go for sensitivity, so like trace detection, low energy detection, low signal detection? Do you go for resolution? So do you have peaks that are often closely spaced to each other and you need to distinguish between different elements? Or do you go for speed? Do you like to collect your spectra quickly? 1 second or 10 seconds is pretty common these days. Your maps could be collected in 2 or 3 minutes is not uncommon at all these days. So which is important to you? And of course, many of you might say all of the above, and that's because we don't like to do compromises. So we want our detector performing all around very well. And I'll give another couple of seconds to have our final submissions for our survey before moving on. So if you don't use an EDS detector, that's okay too. You can think about other techniques. If you use HPLC or even just microscopy on its own, or if you use an atom probe, you can also check which one is important to you. All right. Excellent. This is really nice to see that we've got a lot of good scientists who are out for overall quality and performance. So everybody, the majority, about almost 60% of you go for having your quality performance in sensitivity, resolution, and speed. That's wonderful. I'm guessing maybe we have a lot of people who like sensitivity and resolution together, so you might have had a trade-off there. But all of the above is great to see. And I will encourage you to visit some of our previous webinars where we talk about quality performance metrics of the detectors, and that will touch on some of the finer points of the latest detector technology and how you could achieve all of those all at once. But now we will move on to how you can use EDS in the world of life sciences and biological applications. So there are two main areas that we'll talk about today. And the first area is just the common usage of EDS, so to get a better understanding of what's in our materials that we're looking at. So what elements is it comprised of? And then also the distribution of those elements in the sample. So with an X-ray map, we could look at which elements are present and then also where they find. We could then associate features within their environment or in some cases in finished form. And that is a very common use of EDS. Now, in the world of biological or life sciences, EDS also gives us this really unique opportunity to look into an organism to better understand the morphology and the composition. And this is because we have the ability to adjust the microscope KV to be able to penetrate more deeply in the material, and our X-rays are strong, so they're able to come up through the surface of the material and still be detectable. So we have some nice application examples there as well. So now moving along to a practical example of a hair application, and this is to show off the detector performance a little bit in sort of an unusual sample, but a hair nonetheless. So this was actually collected off of just a single hair from a spider, and the beam conditions we're at 8 kV, which is a good place on many microscopes for these types of applications. Also, having a variable pressure microscope is going to be one of the most important aspects of data collection, and that's because you could work at higher beam energy, you could get that enhanced excitation, but still not damage your sample as much or cause charging or beam damage quite as much. So that the variable pressure capability really lends itself well to life sciences applications. We've also used our silicon nitride detector, which is our Octane Elect Plus, and we have a nice resolution with also a nice 50 eV carbon resolution, which helps give us those low energy peak separation. And again, very quick with a low 7% dead time and a quick 10 live second collection time. With that collection, we're able to confirm that we can see those peaks nicely separated, going down to the baseline and highly resolved. Great sensitivity there, especially for the nitrogen. So a little bit challenging being that it's a small feature, a small hair, but nonetheless, we're able to get great signal. So now we move on to a slightly different application with a marine biological application. Using the same detector, we're able to collect a spectrum of major and minor elements within about 30 seconds. And so in this case, we have some real trace elements below 2 to 5%, and we're able to collect the spectrum and get our data very quickly from it. In this case, we're able to see what's notable is the sodium and chloride, sodium and chlorine, and that tells us that we have obviously salt somewhere in our sample. So we move on to an X-ray map where we're able to look at the distribution of that sodium chloride within the sample. And so this is the underside of a starfish, and you can see the morphology of it, really very beautiful structure. And in the electron image, we're able to see some of those bright areas, which many of us will suspect that they are in fact something different chemically. And with our X-ray map, we're able to confirm certainly that those bright areas are represented in this map by sodium. So the sodium is in this orange color, and you could see it most readily around the edges, the inside section of it. Now, what we can also see with the X-ray map in green, with the oxygen represented in green, we could see that we have some nice topographic information of the starfish. So what appears to be flat in our electron image does in fact have some surface changes that we confirm with the EDS analysis. And again, with the collection within just a few minutes. Now we'll move on to a really interesting softer tissue sample. So this work came about a number of years ago when we had a user school attendee from Montana State, and he talked about a grass called horsetail. And this is a grass out west which has silicon in it. And the reason that silicon was in it, it was two reasons. One is a protective measure. Animals don't prefer to eat grasses or plants with silicon in them because it grinds down their teeth, it wears down their teeth, so they stay away from these. So it's kind of a protective mechanism against predators or herbivores. The next thing that it's beneficial for is giving the plant some structural integrity. So even in the absence of a lot of water in dry climates, the silicon gives some structural integrity to that particular horsetail plant. It can grow pretty tall. So after having met him and talked about this, I did a little bit of research and found out that there were some sawtooth sedge grasses that do also have silicon in them. So I did some Google searches and found these sedge grasses. And these are the things where you walk through a field and they kind of get stuck to your socks, they're a little bit spiky. So I looked for some of these sedge grasses and put them in the microscope. And the first day that I did this, my original approach was to work at low kV. So I put it into my field emission microscope and went to 3 kV, and it was charging terribly. I tried 2 kV, it was still charging. Didn't really see much of anything. Maybe I went a little higher to 5 kV, but probably couldn't get any images at that point. So I didn't see anything. No silicon in my X-ray spectrum, no ability to collect images or maps, and I was a little bit disappointed. And so I left it for a day or two and decided, well, okay, I could do this, so let me try a different approach. And at that point, I then put it in my tungsten variable pressure microscope and then worked at 10 kV. And so now with the variable pressure, I could prevent the beam damage or the charging, but I could also work at higher kV. And when I went to the higher kV, all of a sudden these silicon dumbbell-shaped bodies stood out at me in the image. So right there in the middle of your screen, you could see those bright dumbbell-shaped bodies, and sure enough, that aligns right with the research where they were purported to have these dumbbell-shaped bodies, and sure enough, they lit up when I was working at 10 kV. So just beautiful there. And then you could see some of the X-ray maps showing the silicon and oxygen and the carbon. So usually I try to have the biological colors in green, and then the organic materials in green, and then have the other interesting colors in like a brighter blue or something red that jumps out. So of course, because I did the study and at 10 kV I was able to get really nice signal, then the following year when I had some interns helping out, I said, let's revisit this whole project because it is pretty cool where we can see elements below, that we hadn't seen at low kV. And so why do we see these the signal at higher kV rather than not see it at low kV? And so here is an image of the silicon, and this is, the first image is just at 5 kV. And now I'll move ahead and move it up to 10 kV. And you can see what happens where we now could see the silicon X-ray map showing the dumbbell-shaped silicon. So the bright areas that we saw in the electron image are certainly confirmed now as silicon X-ray signal. And we could move it up to 15 kV with another mouse click, and 20, and so we don't get too much more change between 15 and between 10 and 20 kV. But what we do see, interesting enough, on the area to the left side of the screen, and I'm not doing a poll on this one, it's just of interest. So these areas seem to have the silicon that has come out of the area. And for those of you who are experienced in the microscopy world, you probably know exactly what's going on. And those are the areas that I did a point analysis of. So I did a few spectra collection ahead of time. And when I did that, I left the beam on those areas for probably 10 or 20 seconds. And what happened, it looks like that the beam damaged some of the biological, organic tissue, and the silicon actually spread out from the dumbbell-shaped body. So very interesting there. And now finally looking at the silicon combined with the carbon X-ray maps, so we could clearly see the regular structures of the silicon on the flat surface of the grass. So this is the flat surface of the graph, not inside of the grass, not inside view. Now we'll take a look at the side view of the grass, and these are the thorny spines on the edges of the saw grass, and this is what you feel when you rub your hand down the side of the blade of grass. And so looking at that chemically, we could actually see that those thorns are made of silicon and oxygen, otherwise known as silicon dioxide, and also otherwise known as glass. So I found it tremendously interesting that there is glass in grass, and so the X-ray maps highlight exactly where that is and how we can understand that more deeply. The next example is looking for metal antioxidants in a grape seed, and this is where we've all heard that antioxidants are helpful for us, and so they play a particular role in the body by reducing the oxidative stress on certain areas, certain cells in certain areas of the body. And so we also see that certain seeds or certain plants are high in antioxidants. And so I decided to look further into that. While there's a whole lot of work on wet chemical analysis, HPLC or GCMS, on the presence of antioxidants in plant material, there's very little work done about the actual location of the antioxidants in the plants. So the first part of the experiment was to take some grape seeds and open them up and then look inside. So in this case, I'm at higher pressure, so these were charging quite a bit. So I have higher pressure in the microscope, and I'm also using the backscatter electron detector. And when we use that, then these bright areas jump out at us. I found a high presence of many minerals, namely calcium, in the seeds, but occasionally there are small manganese particulates, and after a longer spectrum collection of about 30 seconds, we have detected a manganese peak, which is the peak all the way to the right of the spectrum. It's just there, just slightly above the background. It turns out it was about 0.3% manganese, and that's when we just placed the beam right on that particulate. What also is probably going on is we're collecting from a wider area or a larger area, and that's due to the spread at the higher pressure in the microscope. But the result is that we're able to actually detect these antioxidants directly within the grape seed. Another very interesting aspect of this is the ability to detect the other minerals that are found at the edge of the seed coat, but are not observed throughout the seed. And the most important one that I found over and over again was calcium. And as it turns out, calcium plays a tremendously valuable role in seed germination. It is a metabolic stimulant and helps with germination when it's present in the soil and taken into the seed itself. It also controls the solubility, so it better allows the seed to take in the water and the other important nutrients to allow it to grow. So to now apply just some cool and interesting facts and figures into some real world work. So the seed industry, the plant industry, the growing industry is so important for all of us who eat anything plant-based, which is probably most of us. So any time that you have an experiment where you want to look at how much calcium is present in the seed, what the impact of the growth rate is, you can use EDS to study the correlation between calcium in the seed and the solubility and germination of that particular seed after any presence or changes in the soil. What I've also found, based on research, is that calcium in the cell wall increases the resistance to some infection of materials, fungus, and viruses. So calcium plays another secondary important function in plant materials as well. Finally, looking at just a few examples inside the seed itself, a little bit of a higher magnification, where we can do point-by-point analysis to look at the chemistry of the bright particulates within the seed. And we see that many of them contain calcium, but not all of them contain manganese, just a smaller fraction of them. Finally, moving on to our last EDS example before moving into EBSD, we'll look at our spice clove in a little bit more detail. So this is one of the smaller features of the larger feature that we saw in the first slide, and we're zooming into just a particular area. And based on my research, I saw that spice cloves are one of the highest food substances with antioxidants. And so I was expecting to find an awful lot of interesting higher energy or higher atomic number materials within the spice clove. So looking at the first electron image at 8 keV, we don't see a whole lot of interest there. And that is kind of consistent with the sawgrass experiment also done. So at lower beam kV, we're not quite seeing everything that we can see. But we go to a little higher beam energy at 15 kV, and now we're able to see some of the porosity inside of this feature, and we're able to see some of the bright particulates. And these are actually below the surface. So I'll go back to the 8 kV, which gives us just really a surface view. And now we're going a little bit below the surface and looking at some of those open pores and also the bright areas, which indicate the antioxidant material. And we go even higher kV at 25 kV, and then we could see right there through the center is where the cluster of antioxidants is found. So throughout the length of this seed area, we could see those bright particulates, and those are confirmed with EDS chemistry to be the antioxidants. So one last poll to get an idea of why we're able to see features at higher kV than we can't see at lower kV. And because in the world of soft tissue analysis or biological analysis, we tend to think work at lower kV because you don't want to damage your sensitive materials. You don't want to burn your tissue. You want to preserve your tissue. That's the common approach. But there is a benefit to the higher kV. And let's see what that benefit is. If you want to submit your answer, I'll move along. Excellent. Glad to see that that everybody was paying attention, and we can see things at higher kV because our electron beam penetrates more deeply into the material, and we're able to get the electron information as well as the X-ray detection below the surface of the material to understand the internal morphology as well. And this is a final spectrum that certainly at 15 kV that confirms that, again, we have those manganese, those small manganese peaks present as our antioxidants. And one slide to throw in that talks about why we're able to gain the benefit of faster processing even at low count rates. And this is high speed even at low count rates. And that's because we have fast processing times of detectors without having to make that compromise in resolution or sensitivity. So early on when we took that poll about which performance parameters are important to you, most people said that all performance is valuable, and this shows why all performance aspects together are important. For example, in the oxygen intensity, even when we're at low count rates, this is just a few thousand counts per second, I think it was 5,000 CPS, and we're able to work at faster process times comparatively. So when we work at 7 microseconds versus less than 1 microsecond, we're able to actually get much better intensity. So lower dead time and higher peak intensity. Now we're just talking a few percent. I think it's a difference between 5% and 0.5%, but certainly we like to have even the small benefits, and you can see by the peak intensity that we are gaining something when we go to the fast processing times, but we're really not losing anything in terms of peak intensity. the other thing worth noting is that many of the applications are used in regulated industry in these arenas. And the regulated industry has some federal regulations for CFR 21 Part 11. This is for data integrity and tracking. And we at EDX are making some huge strides into conforming with those regulations as well, so that any users in those industries can not only reap the benefit of the detector performance, but also with the software. So in the last few minutes of the presentation, I will go through two different EBSD analyses of biological materials, and these were provided by my colleague Renee Declos, who is always after very interesting applications and challenging applications of EBSD. So the first one that we'll study is the EBSD analysis of the nacre layer in a shell. The nacre layer is the iridescent layer, or otherwise known as mother of pearl, and this serves as some functionality within the marine organisms. What Renee did, working with his colleagues at BCube in Dresden, was to look at the Prius images of the inside of the shell. And Prius is a technique within our EBSD offering which allows us to look at an EBSD image rather quickly, but still see some important characteristics, much like we get with a forward scatter detector, except we can see even more than we could see with an FSD. So in your left image, we've got our Prius taken from the bottom. And what it highlights here is topography. So you can see some of that layered structure, that lamellar structure, and you can see the regular repeating pattern of it. When we move into the middle image, we could see some orientation contrast now. And so this gives us an idea of how the grains are differing in orientation from point to point. And finally, when we go to right side, so the sides of the phosphor, we're able to see some atomic number contrast, which is equally important to help describe the material. And now what we do is we take a full EBSD scan that shows the inverse pole figure, or the orientation, for each of the sampling points from the EBSD scan. And what we see is that the lamellar structure has repeating orientation throughout what you would consider grains. So it's not just the lamellae or the individual appearing grains, but rather the grains are looking larger than just the individual segments. And then what he has done is taken grain boundaries, which we see in the center map in yellow. And then the right, the red in the right side shows us where the contiguous grain boundaries are. And we could see a really good correlation between the grain boundaries and the contiguous grain boundaries, meaning that the grains are larger than just the individual layers of lamella. On the right-hand side, we are simply showing a unique grain color map. And what that does is it helps us sync in where there are the same grains. So for example, in the middle here towards the left, there's this large yellow grain. So that's considered a single large unique grain. And when you look at it in the inverse pole figure map, you can see that they have all the same orientation as would be expected. So those grains are larger than you might otherwise think just by looking at the structure. The next thing that we could do is look at the particular character of the grain boundaries. And when we look at the misorientation angles of the grain boundaries, we're able to see that there is a repeated distribution around 63 degrees. And that means that there's a certain favorable orientation that we're seeing over and over again. And this is what I believe to be a coincident site lattice or CSL boundary, and it's something that's more energetically stable. And when we highlight the peak, we're able to track where those grain boundaries are. And if we zoom in a little bit more, we could see some of these certainly do appear to be like twin boundaries, because within the lamellae themselves, we're able to see some small changes. So if you take a look at some of the smaller features, you could see that within each lamella you have a flip in orientation in small areas. So now we'll go through a series of comparisons of our forward scatter image. So this is directly an FSD image, which is a solid state diode on the EBSD camera itself. The FSD has been around for a long time. It's a simple silicon diode. And so it gives us an idea, a little bit of topography, a little bit of maybe orientation contrast of the area of interest. But now when we move to the Prius map, we're able to, or the Prius image, we're able to see now some orientation and even a little bit greater detail to the topography. So we're highlighting more information with the Prius image. And then we go even one step further to look at the image quality map. And the image quality is directly an EBSD scan and a representation of the band contrast. So the higher crystallinity areas have sharper contrast and more detailed IQ. And finally, looking at our inverse pole figure on top of the Prius center image. So we're able to get some of the orientation contrast from the Prius filled in with the orientation color of the inverse pole figure, plus some of the topography all together in one image. And finally, we could even look at the growth direction. And this is really interesting because we see that in the larger area, the lamella area, it's more consistent, but as it goes towards the edge, we have some changes in the growth direction as seen by the pole figures on the right-hand side. And we'll finally just take the last couple of minutes to look at a really interesting fossil example, and that was done by University of Glasgow, and we've had around with our EBSD studies for quite a number of years now, but it is one that really stands out as different. So trilobites are a marine animal. They are now extinct, and they have a hard exoskeleton, and they were one of the first organisms to evolve eyes. And those eyes were actually a part of their exoskeleton. So every time they shed, they would shed that top layer of their eye material. Now, of course, exoskeleton makes us think that we have something of high order. So we have something that is repeated and subject to growth. So the trilobite eyes are determined to be composed of crystalline calcium carbonate, and that we can do by EDS detection and testing. But then what we do with EBSD is understand the structure of those better. So if we look at the arrangement of the calcite crystals, so what we see is, the calcium carbonate, sorry. What we see is when we look at a crystal, when the crystal is highly ordered, meaning that all of the grains are in the same direction, it appears clear to us. Now, if you have a crystal or material that's hazy or fuzzy, that's usually because they have many different orientations, and that type of arrangement would not be a great eye, right? We want our lenses to be clear, and in order to be composed of the calcium carbonate and be clear, they need to have the same orientation for the organism to be able to use them effectively to gather light and interpret their surroundings. And so we look at the comparison between different aperture structures and focal points within an eye in an organism, and we could actually tell the difference between nighttime organisms and daytime organisms because they adapt differently. If we have a daylight organism, then gathering sufficient light is usually not as much of an issue, so you're not optimized to gather as much light as possible. But in a nocturnal creature, it wants to gather as much light as possible from low light situations and focus that available light onto the lenses. So we have a different arrangements between the opening of the eye and the direction onto the lenses here between them. So we use that information to then move into the study of the trilobite lenses. And so this is an EBSD scan showing the inverse pole figure, which also gives us an idea of how ordered the structure is. So of course, when you see many of the data points of the same color within the lenses, you know that that's a highly ordered. So all of the orientations are in the same direction, and that allows it to gather clearly the light into it. what we could also see on the top right area of the lens, the lens on the top right, is that the center area has a very strong order, all in the same direction, but the outside of the lenses are slightly different in orientation. So this is kind of a ring of subgrains. And if we zoom in on that a little bit further, and then we superimpose the actual crystal orientation on top of it, you can see that they're all pointing in the same direction, all to gather the light in and to focus it into a central point, which is really just fascinating. And we could kind of say that that's similar to a compound eye of a modern insect, because the compound eye is all focusing into the same direction. And in the final view of the study, they did a cross-section of the trilobite eye to look at the central core and look at the orientation. So based on the orientation, we see again some of that regular structure followed by an inner core of a different orientation. And we can see the pole figure here that describes the focal, or how all of the outside areas are pointing in towards the center area. And finally, we sample that around the outside of the eye in the cross-section, and we could see the trend of the orientation as we go around the eye. And then we look at how it focuses each lens compared to a compound eye. And the goal is that we're pointing forward and collecting more light, or the conclusion there is that we're pointing forward, collecting more light. Okay, so that concludes our final example of our EDS and EBSD life science applications. In summary, there are a growing number of applications in biological and life sciences where SEM, EDS, and EBSD are providing new information. So we just have to go out there and explore what those applications are, and certainly when we do, we find some really interesting examples. We can use the characteristics of the electron beam to look at both surface and subsurface analyses and understand what's going on inside of our biological material, so even more than we can gather just by microscopy alone. When we pair them with latest detector technologies like the silicon nitride thin window and the fast processing speeds, we're able to get the sensitivity that we need to be able to detect trace amounts of materials, and all the while process them quickly to prevent a lot of beam damage to our sample. So I encourage everybody to explore and discover new applications of EDS and EBSD in the life sciences. I'd like to also send a thanks to Renee Declos, as well as move on to our final slide where we can look at some additional resources from EDAX. Renee has done a great blog on some of these fossil applications, and so please check that out. We also have some movies that will show you more of the detector characteristics of speed and resolution and sensitivity, so you can learn those in greater detail. Our past series of webinars go through everything from EBSD sample prep and biomimetics to detector speed in EDS, so please feel free to check those out. And we have upcoming webinars in November and December with our EBSD colleagues, one of who has a birthday today, Matt Nowell, will be doing an EBSD webinar next month, and please join in to take a look at those offerings. So I'll now move over to some of our questions and answers, hopefully some answers. Oh, great question. What about sample prep? Does it vary from routine preparation? So this question brings back really challenging times for me as a biological student in undergrad, where I prepared a TEM sample based on having to dry it out and mount in wax and microtome, which is really very, very challenging. And nowadays, fortunately, things are a lot easier because we have variable pressure microscopes available to us. And with VP SEMs, we're able to to work at higher kV, but introduced pressure into the chamber so that we're able to dissipate the charging. So biological materials don't conduct the charge of the E-beam away. They retain that charge and then they discharge it at events, but when we have variable pressure, they can regularly discharge. So that beam current that's going into the sample is then released out into the pressure inside the chamber. So sample prep is not as difficult as it was in times past. You just need a variable pressure microscope. And then also, if you do want to cross-section, there are a number of ways of doing it depending upon how large or small your sample is. For larger samples like the spice clove, you can just take a tweezer to poke into the sample. But for finer samples, you would want a fib to mill away a surface of a smaller area within the sample. I've got a question of the, let's see, the shadows on the starfish. Yes, that's a great question. So the electron image on the starfish, and I think I will scroll back because hopefully this, yes, okay. So in the starfish image, the electron image shows a flat image, and we're working with backscattered electron images, which give us a flat look. It's also what allows us for the sodium chloride to stand out, which you know. But when we look at the EDS map, so the EDS detector comes into the chamber at a side entry, at an angle entry, whereas the backscatter detector is flat above the sample, so it gives that flat appearance. But the EDS detector is subject to topography because it has that angular view to it. So anything that's shadowed or anything that's pointed kind of away from the detector is going to appear shadowed, and that's certainly what we see there. We have a routine called CPS mapping, or counts per second mapping, and that's really a beautiful map enhancement capability that shows the distribution of the counts per second at each pixel and highlights those that topography. But what it also does is allows us to non-highlight from that topography. So in times we don't want to see topography because that interferes with our chemical signal. So for example, where we see less green, doesn't mean that we have less oxygen there, it just means that we have less X-ray counts per second. So our CPS normalization feature will normalize or enhance the areas of low count rates while at the same time decrease the highlighted areas in that as well. Let's see, do I put on a conductive layer? Nope. These days we're super lucky to have the variable pressure, and that eliminates all need to do a conductive layer. And this goes far and wide for biological samples, everything from the plant materials that we saw to the biological tissues that we had on there. Hair is pretty easy. The one that was a little bit more of a challenge was the grape seed. So that was higher pressure than I normally use. That was about the highest pressure, I think about 60 pascals, but we're still able to get nice looking images, good signal, and all with just the variable pressure of the scope. No need for that. Oh, thanks, Dave. Let's see. Which are the best parameters for improving quantitative bidimensional map? Oh, really, really interesting and good thinking. So for those of you who are not familiar with quantitative maps, quantitative map is going to have a sampling point. Each pixel in the map is going to be quantified and give a display of the atomic percent or the weight percent of a material. This is very helpful because sometimes when you have low energy materials or low atomic number materials, the regular map, the raw data map, suppresses those low energy or low atomic number signals. And so if you have the presence of an organic material in a heavier material, you might not see that as well. So the question is, which are the best parameters? I think the single best parameter is going to be improve the speed of your process time. The process time is known interchangeably as amp time or pulse processor time or peaking time. So all are kind of used, and nowadays our fastest pulse processor time is 0.12 microsecond. I wouldn't go that fast. I would say the best balance is going to be somewhere at about half of a microsecond. We've got a 0.48 microsecond amp time, and that's quite fast. Typically on our detectors, we're still able to be in the low 130s, 131 EV or so with a half a microsecond process time, and this would be compared to the slowest process times are usually around the 8 microsecond process time. So you're going to speed up your collection, you're going to get more counts through, more throughput, and more counts throughput is more statistics. More statistics translates into better quantitative material. So for all of those data points that you gather in a map, you want to have the highest input count rate, and you could do that with your pulse processing time. Let's see. And yep, you can also reduce the Bremsstrahlung for quantitative mapping. How? The way that you, so yes, you can do that, and how we do that is with a map that we call the net intensity map, and that does, for each pixel, it does a peak to background fit, and it subtracts out the background, and that's going to give you the better signal above noise for your map display. Oh, yes, for increasing only the nitrogen counts of the hairy house spider, what are some of the tricks? And one of the best tricks for low energy detection is to tilt the sample. So a tilted sample reduces the exit path or the mean free path of the X-ray as it's escaping the material. You have to tilt towards the detector, but when you do, it means that the X-ray travels less distance through the sample and a trajectory to get towards the detector. And less path through the sample means less opportunity for low energy X-ray like nitrogen absorption. So better exit of the nitrogen, better detection for nitrogen. So if you can tilt your sample, or in the case of a highly irregular sample like a spider, you would just want to find an area, the topography where the hair is pointing towards the detector. I've got a question about correlative studies with the nacre material, and Hugh, I would definitely suggest that you reach out to Renee Declos. I think you guys would have some great conversations about putting your skills and techniques with Atom Probe and TEM together. That would be very interesting. Okay. On the hair spectrum from the polymer window showed digitization. I think what that means is that the spectrum appeared to be rougher. And the reason that in the polymer window the spectrum looked rougher was because it had lower signal. So the two spectra were scaled on the same scale. Because the polymer window had less intensity, it had to be scaled up, and that's why the digitization is greater appearing in the polymer. Somebody mentions doing an analysis using an environmental SEM. Definitely the environmental SEM is going to give you some really nice advantages there, as well as just the traditional variable pressure or low vac SEM. Okay. The question of sample prep for the shell. Did you grind or polish the shells before collecting EBSD? And I can't say for absolute sure because I didn't do the work myself, but I'm quite sure that there was no sample prep on that. I believe that was just in the native state. So good question, and if you'd like to have us reply by email, I could ask Renee on that one. Oh, yes, here's a good question. Could I subtract the oxygen coming from the background? Yes, certainly. You can normalize oxygen. You can subtract oxygen from one spectrum versus another. You could subtract X-ray maps the oxygen from one versus the other. So any of those are possible. Now the environmental SEM or the pressure of the SEM, it depends on what gas you're using, which you might want to subtract. Many of them are just ambient and others are nitrogen, so that would impact how you do the comparison of the map or the spectrum. Okay. What is the max pressure you still trust your EDS data? And does the higher pressure affect the detector? Really good question. So the max pressure that I would use, and this is just kind of, I'm just taking a feel for how I operate my scope. I would say 60 pascals would be about as high as you would reasonably go, maybe a little bit higher than that. We do also offer a mathematical routine called VIP Quant, and what that does is you take spectra at 2 different pressures, and then you can back extrapolate. You plug both of them into the program, and then it extrapolates what the spectrum would look like at high vac. So I would go up to about 60 pascals, but anything more than that, if I was doing spectral analysis or quantification, I would incorporate the VIP quant routine that we supply. And does the higher pressures affect the EDS detector? Great question. The silicon nitride window is very inert to any of the gases or humidity that you would have inside of the microscope environment. So even the polymer window should be fine too, but the silicon nitride window is commonly used as a barrier layer in microelectronics. So that's going to be your surest chance of integrity of the detector in those environments. Okay. I think we've gone through most all of the questions, and I will conclude our webinar now. Please feel free to reach out to us for any further information, and as always, thank you very much for attending. I'm happy to work with you. Thank you.